त्यागी ज्वाइन किया है ज्वाइन किया ना
हाँ इनकी त्यागी है in YouTube there is one person so दो लोग हैं
Anuvidya she's there. MK Tyagi is there. There is shows here.
yeah in YouTube it is he is there
He's in YouTube.
yes
And Anuvidya, where is she?
Anuvidya is our channel no
Oh, channel. Anuvidya.
you in the chat box you write register your part this one attendance please register your attendance
yes
You can start.
I think we can start I don't
Okay.
you're already
So,
invited.
all right. So good afternoon, everyone. And during the last two lectures, we actually we covered biomolecular NMR, certain aspects, and we covered applications to nucleic acids. We started from the basic units of nucleotides and then how they are connected, how the nucleic acids are done, and how to use NMR to characterize the structure of nucleic acids. We discussed the various things, the DNA, RNA, different types of structures, and what are the various characteristic features and how they show up in the NMR spectra. 1D spectra, 2D spectra, even the proton spectra. So now onwards, we will be focusing on proteins, shifting our attention to the next kind of biomolecules, namely the proteins. And what are the NMR methods? This is a kind of a very important slide, very informative slide. We'll go through it slowly. For proteomics, structural biology, and protein folding research. Proteomics deals with the proteome. So the proteome, what is proteome? I've discussed in the very first lecture. The proteome is a group of proteins, all the things which are proteins which are expressed inside the cell. There are approximately 100,000 proteins of different sizes. And to characterize these proteins is the area called as proteomics. And structural biology relates the structures with the biological functions. How do we understand biological functions? Using structures, that is structural biology. Therefore, structure determination is a prerequisite for this. And protein folding is at the fundamental level, how the proteins are formed, what is their structures. Okay, how do they achieve their particular structures when they are synthesized inside the cell, after they are synthesized inside the cell. So that constitutes the so-called protein folding area. That is all summarized here. You can see this, the focus on the different parts of this slide. The first slide, So here the protein world consists of so many things. This is the so-called ribosome here. This is the ribosome where the proteins are synthesized. Okay, we talked about it, messenger RNA coming in, messenger RNA carries the impression about the protein amino acid sequence, and in the ribosome the protein is synthesized. As the protein chain comes out, it is in some sort of an unfolded state. In fact, I must say that some little bit of this I had covered in the previous set of lectures also towards the end, in the applications and some of the applications of NMR. But here we are going to go into the great details so that you can practice and study these things for your own research. Now, so when the protein chain comes out, it is actually an unfolded state. So this is the protein chain. Unfolded state is schematically represented in this manner, the single chain. This is a slide which I've taken from Chris Dobson's one article in Nature in 2003. And very elegantly described the processes that are happening inside the cell. The protein is in unfolded state. And what is going to be its fate? So there are many fates possible for it. One thing is it may get degraded. Suppose it is toxic to the cell, then it may get degraded. There are mechanisms inside the cell which will recognize things as which are toxic to the cell and they will start to destroy them. And it may get degraded. And then, or it can also get into an aggregation, so forming a disordered aggregate, which is this disordered aggregate, or it can get degraded into smaller fragments, and the smaller fragments again can aggregate to forming a big species which is not soluble. That sort of things can also happen. Or these degraded products can form an ordered aggregate, so which is called the prefibular species, and they can further aggregate into different kinds of a structure which is called as amyloid fibrils here. Alternatively, this unfolded chain can try to go to the folded state, the so-called native state. It can go to the native state, but the process of going to the native state is in itself a subject, and that is called as the protein folding problem, protein folding research. There can be certain kinds of intermediates that may be formed. They may be stable intermediates over there, partially stable or transiently formed, but it may go through these intermediates. And if there is an intermediate also can aggregate into form of prefibular species and ordered aggregate, or it can also go into disordered aggregated state as in this. And these ones are not useful. When they have a certain structure, they will be functional, and then that leads to our structural biology research. But if there are disordered aggregates and disordered species, they can actually contribute to as a kind of a pathogen. They may not be useful. And for example, this amyloid fibril which is formed, this is typically the kind of a structure which is formed in all kinds of neurodegenerative diseases. Your Alzheimer's disease, Parkinson's disease, Huntington disease, all of these things arise as a result of formation of such kind of amyloid fibrils. And eventually it may be a good idea to know that the fibril species is actually the most stable species. That eventually every protein will go into that at some stage or the other. So it is related to aging as well. As you grow older and older, the system actually goes towards a amyloid fibril stage, so neurodegenerative stage. And once this kinetic intermediate is formed, then it may further go into a native state called as the folded protein, which is called the native state. So therefore, there are many channels open for the synthesized protein to go into that. And what drives this? This is actually a kinetic phenomenon. So this is the kinetically, whichever one is faster, it will go into that. So you can go into the trapped state. Thermodynamically, the state which will appear is finally the fibrillar state. But these are all transiently formed. The native state is also a kinetically driven species. And the native state is the folded state, which you all know, and they are responsible for biological functions by and large. Now, this native state can also form an oligomer, and this happens in many situations. Many proteins self-assemble to form an oligomer. All of the chaperones, for example, are a classic example of that. GRO-L, GRO-S, these are all aggregates, systematically formed aggregates, or so-called assemblies. We may call this assemblies of several proteins. Or the viruses, for example. The viruses on the surface have several copies of the same protein, and which are organized in a very systematic manner. And those are the big assemblies. So such kind of things can also happen, or it can form a fibril, or it can form a crystal. So crystal is basically a technique. You crystallize it to study the structure of the protein. Now the crystallographers will want to do that, and the NMR spectroscopists will try to focus on this protein native state here, or on this state here, or on this state here, or on this state here, to understand the phenomena, the process that happens in the biological cell. So this has to be done for 100,000 proteins inside the cell, and that is the proteomics research, structural biology research. This is not an easy job. So it is going to be an extremely complicated and competitive process to understand these things in a very rapid manner and in a way that you understand the biological function. Therefore, there is a continuous explosion of methods in NMR and also in other techniques, even in X-ray crystallography, continuous explosion of techniques to do things rapidly and as fast as you can. And that is an important concept, important aspect of doing structural determination in NMR as well. The methods development happens as a consequence of this. As we go along, you will see that various kinds of situations will appear. There is one other situation which is not listed here, and that is called as the IDPs. IDPs. What are these? These are the intrinsically disordered proteins. These are proteins which will never fold to any particular native state. These remain unfolded or random coils, or they may have some partial structure here and there, but these are IDPs. But earlier it was thought that these IDPs may not be important. It is only the native state which is relatively important for the functional point of view. But today we know that IDPs play a very important role in the function of proteins, because these same IDPs can adopt, a particular IDP can adopt different kinds of structures when they interact with different sorts of binders. Small molecule binders, big molecule binders, RNA binders, or various kinds of drugs which are antibiotics. All of these things actually adopt different kinds of structures depending upon the binding partner. And that's why these have become very important. And not only that, even in the folded proteins, it turns out that there are many areas, the so-called loops inside the protein areas, and these are flexible. So IDPs are very flexible. They don't have unique structure. They are very flexible. Okay, and there are loop areas in the proteins which are also flexible, and these are also important for the function point of view. Therefore, it is necessary to understand the roles of disorder in the proteins. The role of disorder and the role of dynamics in the proteomics. So all of these things are quite important in this field. Now the protein folding itself is an important subject. How does the protein fold? Whenever it goes to the native state, that is here. The unfolded protein belongs here. This is the synthesized protein which is at the top here. And this has multiple conformations. It rapidly exchanges between many different conformations. And on this axis, we have the energy. This is the unfolded state. The energy goes up like this. This is energy. And the protein has to adopt a state which is stable. So it has to come to a lowest energy state, and that is the so-called native state. In the unfolded state, because it is flexible, there are many conformations possible. In a chain, we will see that how that happens, various kinds of conformations, how they are possible. But there are millions of conformations that are possible for a chain of, let us say, 100 amino acid residues or 200 amino acid residues protein. So this unfolded state, how does it pass through? So to a such a folded state in a very rapid manner. Sometimes it is even microseconds. Typically, if one has to calculate using the degrees of freedom, it may take an astronomical figure, number of years it can take. However, the proteins fold within a matter of microseconds, milliseconds, sometimes seconds. So how does that happen? Is it a smooth path? Is it a two-state model? Or there can be, when there is an intermediate, if it is a stable state, then it is not a two-state model anymore. Two-state model means directly from unfolded state to the folded state. There is a two-state model. But there are some intermediates, it is not a two-state model anymore. Depends upon how many intermediates are there, you can have many state models. So therefore, all of these things have to be represented. And that is schematically represented in this so-called funnel, which is called as a folding funnel. There are various barriers here. The protein chain folds from here, and folds from here. All of them can have different pathways, and there are many barriers. What you will see is an average of all of these things finally. But you need to understand what sort of ensemble average happens, what sort of elements are present in this ensemble. We need to characterize all of those things. This you can do when you do your experiments as a function of time, the kinetic experiments. So therefore, you see the targets, our research problems are very well described in this particular slide, on this slide. Okay, so now we have to go back to the desk and see what is it that we want to do. What are proteins made up of? I described this previously, but we will now do it in a little bit more detail. And proteins are made up of amino acids. There are 20 different types of amino acids, and the simple amino acid has a formal structure like this. This is the nitrogen here, the NH2 group, and this is the C alpha group, and then there is a side chain attached here, and then the carboxyl group here. COH. This is an amino acid. And there are 20 different types of amino acids. They vary in this R1. This so-called N-C-alpha-CO, this is the backbone. This is so-called the backbone. So this is amino acid. This is R1 is the side chain, and there are 20 different types of amino acids. And this is called as the N-terminus, and this is called as the C-terminus. Okay. Now, if two amino acids join like this, the carboxyl group is here, COOH, and the NH2 is here, and this OH combines with this H here, combines with this, actually together one water molecule is eliminated, then it will form a bond between C to nitrogen here. And that is what is found here. This is called as a peptide bond. This peptide bond creates a dipeptide. One water molecule is gone. Okay. So now this peptide bond has a very unique feature. You see, this is a carbonyl group here, C double bond O and the NH. But this will have a resonance structure in the sense that this CN bond acquires certain amount of double bond character. So because of the double bond character here, free rotation around that bond is restricted. It cannot have a free rotation around that. Whereas the other two angles which are there, this is this angle and this angle, rotation about this bond and these bonds is freely possible. So these two rotation angles determine the relative orientations of one amino acid with respect to the other. This one. This peptide bond is planar. So this is either cis conformation or trans conformation. So here what is shown is a trans conformation, C double bond O NH, and this is a planar geometry. Okay. So this torsion angle is fixed. We can say it is 180 degrees, the torsion angle, and these other two torsion angles can be very different. And that is schematically shown here in the with this stick diagram here. So one water molecule is removed, and you form here NH2, CHR, CO double bond O, NH, okay, then things like that. So this is one amino acid residue, this is the next amino acid residue. Typically, the numbering of the amino acid residues goes from the N terminal to the C terminal. So if I say this is number 1, this will be number 2. And if it goes further, then you will have number 3, number 4, and so on. So numbering of the amino acids in a peptide or a polypeptide chain goes from the N terminal to the C terminal. Now, what are these various R groups? I said there are 20 different kinds of R groups, and these are very different structures. And these are classified into different categories. You have the neutral amino acids, or acidic amino acids, or basic amino acids. Okay, and by and large, there are neutral amino acids are more in number, and these are alanine. And okay, once again, so far as the stereochemistry is concerned, there are two kinds of configurations which are possible, and they are called as all the chemists know L and D. And here in nature, we only have L type of amino acids. And all of these are naturally. Why is this so? One doesn't know. Why the nature picked up the L amino acids and not the D amino acids? One does not know. People try to synthesize peptides with D amino acids, and actually they turned out to be very unstable, but in some situations they are also formed. So therefore it is possible that it is a process of natural selection which eliminated the D amino acids in the natural proteins, and you only have the L amino acids in the protein. So the neutral amino acids are L-alanine, okay, all L's only. I will say it only: alanine, asparagine, cysteine, glutamine, glycine, isoleucine, leucine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, valine. These are the neutral amino acids. They don't have a charge, in the sense that the side chain doesn't have a charge. And among these, there are a few which are hydrophobic in nature, in the sense in the side chain they have the rings, which are those. This is the tryptophan here, and tyrosine here, and there is phenylalanine here. These three are aromatic amino acids. There is one more which is partially aromatic, and that is the histidine. The histidine is also included in the basic amino acid because it contains a positive charge in the side chain. Partially positive charge, so it has in the side chain. Therefore, it is included in the basic amino acid there. And acidic amino acids are aspartic acid and glutamic acid. These are the two acidic amino acids, and the basic amino acids are L-arginine, histidine, and lysine. Okay. Now, there are several residues which have, the side chains of different lengths. Some of them have 2 carbons, 3 carbons, 4 carbons, and some of them also have the CONH2 group. There are 2 amino acids which are the CONH2 group in the side chain. That is glutamine and asparagine. These two have CONH2 group here, okay, CONH2. And they stand, why I'm pointing out all of these things is, they stand out distinctly in the NMR spectra. CONH2 group when is present, all when is the basic amino acid, the side chain has a positive charge, acidic amino acid there is a negative charge. So all of these things are present in your, they will show up in the NMR spectrum. So when you analyze the NMR spectrum, all the structures of the amino acid should be there in your head. So therefore, you can immediately identify the chemical structures of the individual amino acids should be there in the head. Only then you can analyze this complex NMR spectra without much problem. So, and it will automatically come as you go through the spectra one again and again and ponder over it and think over it as to what side chain it must be. We will see those things, how this will come. Okay, so we will be coming, referring to this again and again and again. Okay, now we can describe the structure of a protein at 4 different levels. One is how the amino acids are joined. 20 different amino acids, they are all joined head to tail, and that is the so-called primary structure, like a beads on a string. Okay, so you have these various amino acids which are joined head to tail, and this forms a polypeptide chain or the protein chain. When it is a small, we say it is a polypeptide, and when it is large, because they call it the protein. And now these ones individually, they can have a certain relationship with respect to the stereochemistry, how they are oriented with respect to each other in the polypeptide chain. What sort of a structure they can take, individual amino acids, to form a kind of a long structure. And you see there are two kinds of structures which are known, and that is alpha helix and the beta sheets. Okay, two kinds of structures which are formed. How they are formed, we will see. So this is the so-called secondary structure. This amino acid sequence is called as the primary structure, and the kind of a secondary structures which are formed, how they are arranged with respect to one another in a systematic manner, that is called as the secondary structure. Now these individual helices and the beta sheets, they can fold and then interact with each other and form a three-dimensional global fold, and that is called as the tertiary structure. Now tertiary, there is on the next level is the quaternary structures. Multiple species can associate themselves, a dimer can be formed, or the four-strand structure can be formed, or many the species can aggregate to form a structure in a systematic manner. That is called as a quaternary structure. Typically, you will find all of such things in very complex systems like nucleosomes or virus particles, as I mentioned. In all of those things, you will have this quaternary structure. Typically, the enzymes and things like that, or small proteins, they will have the tertiary structure. We'll be dealing with the tertiary structure most often when you deal with biological function. Okay, this is more details about the structure. This already I talked to you, primary structure. Now the helices are formed like this. You look at the helix. The chain goes from in this manner. This is the right-handed helix here. It goes like this, the right-handed helix. The NHC alpha CO, NHC alpha CO, which is the chain is coming. Let me look at how the chain is going. This NHC, okay, the chain is coming from here. NHC alpha CO, NHC alpha CO, NHC alpha CO. So the chain is coming in this manner. Okay, N terminal is here, the C terminal is there. Now in this one, this kind of a helix is stabilized by certain hydrogen bonds. Hydrogen bond, there is a NHC alpha CO, NH2CO. This is 4 residues after. This hydrogen bond is formed systematically. Every carbonyl is hydrogen bonded to the NH, which is 4 residues down. Okay, and this way you have a series of hydrogen bonds which are running parallel to each other, parallel to the axis of the helix. Okay, you see so many of those ones. These hydrogen bonds provide stability to the protein structure, to the helical structure. So by and large, naturally, the helix is the dominant structure in smaller species. Of course, there can be other kinds of helices, other kinds of structures as well. Now the beta sheet is formed in this manner. Two chains interact with each other. Here there is only one chain. Only in one chain, there is only one chain in the case of alpha helix. In case of beta sheet, there are two chains running either parallel or anti-parallel. How are they running here? Let's look at this. So this chain is running NHC alpha CO, NHC alpha CO, NHC alpha CO. So this chain, the upper chain is running like this. And how is the lower one going? NHC alpha CO, NHC alpha CO. This lower chain is going like this. So these two chains are going in opposite directions. These two chains are going in opposite directions, and the two chains are held together by these hydrogen bonds between the carbonyl and the amide protons of the chain. So the CONH, CONH, like that. Okay, this one is going on the other side, CONH, NHCO, and so on and so forth. So this forms an this is called as an anti-parallel beta sheet. Now, whether do you need two polypeptide chains to form this, or it can have within the same polypeptide chain. It can have within the same polypeptide chain. It just means that the protein chain has to go in a particular direction. Then it has to take a turn. It has to take a turn and come like this. And the turns will also have a secondary structure. There are two kinds of turns which are described here. One is called the type 1 turn, and there is one is the type 2 turn. Two types of secondary structures are formed here in the turns as well. Now how the chain is going here? So this is NH alpha CO, NH alpha CO, NH alpha CO. It goes in that manner. Okay. So the chain is going in this manner. Okay. This is the N terminal here, and this is the C terminal there. Now, what is the, and the second one is this way. Here also the chain is going like that. This is the type 2 turn. NH alpha CO, NH alpha CO, NH alpha, it goes like that. However, what is the difference? The difference in this is, notice here, in this structure, the carbonyl is pointing inside, the NH is pointing outside. In this structure, the carbonyl is pointing outside, NH is pointing inside the turn. So this happens because of the combinations of the torsion angles which are present here, which we will see later. So there are different torsion angles which are there, and different combinations of these torsion angles lead to these different kinds of orientations of the groups, and that leads to type 1 and type 2. Now here itself the various torsion angles are indicated. So this one, for example, this is NHC alpha CO, NHC alpha CO. And this refers to this particular kind of a type 2 turn. This is a particular, as an example it is shown here. The carbonyl is going out, NH is here. Now, the orientations of these ones are determined by the torsion angles. This residue, I plus 1, this is the I plus 1, I plus 2, I plus 2, phi I plus 2, psi I plus 2. There are two torsion angles per residue. These are N to C alpha bond, around the N to C alpha bond. Second one is the C alpha to CO bond. And that is, both are single bonds there. Both are single bonds. The peptide bond is not, it doesn't have rotation, that is planar, and therefore that remains in that manner only. So these two torsion angles per amino acid residue dictate the relative orientations of one amino acid residue with respect to the other. Okay, these torsion angles, of course, it will go on like that. So therefore, every residue, there are two torsion angles or degrees of freedom to determine the structure of the molecule. So if I have 100 amino acid residue, there will be 2 to the power 100 degrees of freedom. So how to search in those degrees of freedom? Each value, each angle can go to 0 to 360 degrees, or sometimes one says as minus 180 to plus 180. That sort of a nomenclature also one uses. And these ones have to be determined, and that is the big challenge. And that actually comes in the case of protein folding problem or structure determinations, as we'll see. Now, in this, so it is indicated that the chain is going like this and the chain is coming out like that. The distance between the two R groups, the C alpha carbons, C alpha atoms here, is about 7 angstroms in this particular case. And that will be different here. So all of these will reflect in the kind of a structure, in the kind of NMR parameters which we will see. Okay. Now, the third structure which I mentioned, that is about the secondary structures, how they can be formed, and the tertiary structure also already indicates this sort of a formation here. Now, you see the chain is going in a particular way. You have a helix here, and then of course there is a helix here, and then there is a helix here, and there are loop areas here. And these loop areas constitute the so-called turns and all of those ones. Okay, these so-called loop areas. And loop areas are quite flexible. Okay, so therefore, a combination of the helices and how they stack with each other with respect to each other, that determines the tertiary structure of the molecule, or the three-dimensional structure of the molecule in space. And quaternary structure is the association of different kinds of tertiary structures. Okay. Now, I showed you earlier an anti-parallel beta sheet. Now, I also show you a parallel stranded beta sheet. So, this was the anti-parallel beta sheet which I showed you, and here I have the parallel stranded beta sheet. See, why is it parallel? So, look at the strands. This is NHC alpha CO, NHC alpha CO, NHC alpha CO, NHC alpha CO. This is running in this direction. And this chain is also running in the same direction. Okay, NHC alpha CO, NHC alpha CO, both are running in the same direction. Okay, and now the hydrogen bonds are now between different kinds of atoms. Of course, it is the same NHO hydrogen bond still, but their orientations and the structural features, the characteristic features will be different. So that is how it is indicated. The hydrogen bond is indicated by this here. Now, in a particular protein, you can have a combination when there is a sheet formation. Suppose we have with several strands are stacked adjacent to each other, you can have a combination of parallel stranded as well as anti-parallel strands. So therefore, you form a sheet here. See, in this sheet here, you can see there is a anti-parallel beta sheet here in this one, another anti-parallel beta sheet here, and here there is a parallel beta sheet. Okay, the chain can fold in a way and come back. For example, the chain goes like this and comes back here, possibly. Okay, and then there is an anti-parallel beta sheet with the chain going NH, alpha, CO, so it will fold like this and come. The chain is going like this, it will fold and come and go like this, and this is once again it will fold here and come like this. So I can draw here, this fold here, this fold here, and now, oops, what happened?
Lose connection है क्या?
As the extension board का problem. Why?
Okay, so this is coming like this. Now this chain is going like that. Now this is another chain which is actually coming in this manner after several kinds of folds probably, and this is coming in this manner, and now these two form a hydrogen bond here as well, these chains. Now here these two are anti-parallel beta sheets, and this is the parallel beta sheet. All of these can form together in one particular structure. So this kind of a complicated structures are possible in real life. Okay, now the problem was to determine what kind of torsion angles are allowed. So we said so many hydrogen torsion angles are there, 2 degrees of freedom. And this was a challenge which was actually sorted out by J.N. Ramachandran. He calculated what are the possibilities of torsion angles, the so-called phi and psi torsion angles. Here this structure is indicated here, NHC alpha CO, NHC alpha CO. Two torsion angles are there, phi and psi. NC alpha torsion angle is called as phi, and C alpha CO, this bond torsion angle is called as psi. So alpha carbon, nitrogen, okay, carbonyl here. So we focus on these ones here. He took a dipeptide, created this, and he calculated the interactions between the various atoms here in this. For various combinations of phi and psi, various combinations of the values of phi and psi, and he determined for what values of phi and psi there are steric clashes. If there is a steric clash, this will be very high energy, and therefore it will not be allowed. Yes, I will just answer this. Sorry.
Yes.
Hello? So he calculated what possible combinations of phi and psi are not allowed, because if there is a steric clash, that is not allowed. It turns out that in this two-dimensional plane, where the torsion angles phi is here, psi is here, is combinations of these goes from minus 180 to plus 180, minus 180 to plus 180. A large portion of this plane is not allowed. Okay, this entire plane, all this blank area, which is not allowed. It is only in this red and orange. This is the areas which are there. The red and the orange areas, here there is allowed. These energies are less than 5 kilocalories per mole. And the peptide, he is talking about the conformational energy of the dipeptide. Look at the total energy of the dipeptide. Okay, as a function of this phi psi torsion angles. If it is less than 5 kilocalories per mole, then it is practical to accept that. It will be present. Otherwise, it will not be present. It is not possible. So this is simple Boltzmann statistics, e to the minus delta G by RT. So if we have delta G as more than 5 kilocalories per mole, then that is not conformation. It will not be possible. And in this ones here, you have all of these allowed. Now it turns out that if you build a chain with the torsion angles which are present in this repetitively, one after the other, one residue to the next residue, you use the torsion angles which are present in this area or in this area, you will form a particular kind of a secondary structure, and that is the helix here. So you can have an alpha helix for this thick red area, and there is a slightly expanded area which is called as the T310 helix. So it's a little bit more allowed for the 310 helix, and if it is in this area, then you form a alpha helix. So this is alpha helix area, this is 310 helix area. In this area, if you see the thick area here, and that is called as the antiparallel beta sheet or the beta sheet. And there is also polyproline helix, which is also present here. And the collagen helix, this is the triple helix actually there. And those ones also falls in this area, all of these. And the type 2 beta turn is here. The parallel beta sheet has this combination of torsion angles. The antiparallel beta sheet has this combination of torsion angles. Okay, and polyproline helix, therefore you can see, depending upon the combinations of the phi and psi, you have different kinds of beta sheets also formed. You have different kinds of secondary structures formed, antiparallel or beta sheet, or there is a type 2 beta 10. And if we have for the particular kind of a thing, you also have a helix formation. This happens only for the glycine residues. You have some kind of a thing. And extended region, this goes beyond, and then you have something here. So the by and large, most portion of this two-dimensional plane is not allowed. So therefore, if you determine a structure, whether it is by crystallography or by NMR, it has to satisfy these constraints. If it does not satisfy, then it is not a correct structure, you made a mistake in the structural determination, or there is a strong reason which provides stabilization to that combination of torsion angles. So therefore, you will have to provide that answer. Okay, so now with that introduction to the structures, now we turn to the NMR applications. How do we determine the structures by NMR? Now, first of all, to see whether you want to synthesize a protein or get a protein, whether the protein has a structure or not, that is the first thing you have to see. So, what is the characteristic of a protein which has a folded structure? And the top portion, the top spectrum here is indicative of a protein which is well folded, and the bottom portion is unfolded. What does that mean here? Why do you have so many different things? See, there are NH, alpha, CO, depending upon the side chains of particular amino acid. So all the atoms will have particular kinds of chemical shifts. NH, C alpha, CO, or the NH proton, or the alpha proton, or the beta protons, they will have particular kinds of chemical shifts. Suppose there are 10 lysines in your protein chain. Now, if the protein is unfolded, all the 10 lysines will be equivalent. All the 10 C alphas will be equivalent. All the 10 C betas will be equivalent. Therefore, the chemical shift spread will not be there. Similarly, for 10 arginines or 10 glycines or whatever amino acid you are taking, if there are multiple copies of those, they will all have the same chemical shift. Every proton will have the same chemical shift if the protein is unfolded. If the protein is folded, the different residues will have different environments. And because of that, the chemical shift degeneracy will get lifted and you will have a great spread. This is the NH region here. This is the amide proton region here. And this is the C alpha region. And all the other thing is the aliphatic region here. All the methyls and methylenes and all of those things will appear in this area. So these are the NHs and the NH2s. Exchangeable protons appear in this area, this at least downfield areas. And you have around the water. This is the place where the water is. This is the water. And around that area, you have the C alpha protons, then go up further, then you have the C beta protons, and then the gamma protons, or the delta protons, and the methyls, all the way up till here, there's the methyls which come there. So therefore, if there is a good spread in your one-dimensional spectrum, proton spectrum, then you know, okay, this protein is well folded, then you would like to determine the structure of that. If it is not folded, of course, there is no structure there. There is no question of determining the structure. Then, of course, you focus on some other things like the dynamics of the protein. How does it get folded when it binds to certain partner and things like that. Okay, so this is the first investigation. You record a good NMR spectrum and see if it is a properly dispersed spectrum. Okay, now you go to the next level to identify these various kinds of things. What are the regions? What regions correspond to what kind of protons? So you have here backbone protons all the way from here to here. Then you have the aromatic protons here, the side chain NHs which are here, and then the H alphas here, and the aliphatic either the CH2s and the gamma protons, alpha, beta, gamma protons, and then you have the methyls here in this coming in this area. Okay, and now if you remember in the very first slide I also indicated the side chains, the nomenclature of those. I will come back to it again. The nomenclature of this is the C alpha. On the side chain, you go to the side chain, you go to the C beta, C gamma, C delta, and so on. Accordingly, the protons are also labeled as H alpha, H beta, H gamma, H delta, and so on. So you see, this is a well-folded protein, which is all the way goes up to 10 ppm, beyond 10 ppm, okay, and also beyond 0 ppm on the other side. So therefore, this is indicative of a very well folded protein. Okay, now I come to this nomenclature once more for the side chain nomenclatures. So you have these various side chains. Only the side chains are listed here. The proline has a closed ring side chain. The glycine has no beta protons. It has only alpha protons, the 2 alpha protons. Alanine has a methyl proton, CH3 on the side, and that is called as C beta. And arginine goes all the way from C beta, C gamma. I'm not writing the C alpha in these ones here, okay? C beta, C gamma, C delta, N epsilon, and it has this other NH2 group here and the CNH. This is quite a gonadine, it's called a gonadine structure group. And yeah, then you have the asparagine, as I said, it has the CONH2, and this goes up to the beta and the gamma. Okay, gamma it goes. This NH2 will be, you will see the implications of that in the HSQC spectra later on. And aspartic acid, this is the acidic proton, acidic amino acid, there's the COOH group in the side chain. Cysteine has a sulfur in the side chain. So this distinguishes it from all others. Cysteine is a very unique thing because it can get oxidized, it can form disulfide bonds, SH become an SS bond, and then of course it can really change the structure of the molecule, the cysteine. And it is also used for various kinds of modifications in the protein structure by attaching spin labels and things like that. And the glutamine also has a CONH2 here, goes beta gamma, C delta, CONH2 there. And glutamate is again a acidic group, COOH here in the side chain. And this is the histidine. Huh? Any problem?
No, you continue.
So histidine is a side chain here. Now here there can be charged. A particular amino acid, a nitrogen can get charged. So therefore this is included in the basic amino acids there. This is the closed ring. So this closed ring histidine. And other side chains, isoleucine, these are all aliphatic side chains. You have long side chains and they are methyl groups. So methyl bearing amino acids are a unique category in themselves because those ones can be investigated separately. There are many definite techniques which have been devised for those. And the leucine also has side chain, the methyls there are 2 methyls. And the lysine is again as a side chain, is a positive charge on the NH3, and therefore the C epsilon proton here, C epsilon carbon and the H epsilon protons appear very distinctly. We will see those chemical shifts. Okay, and then you have these aromatic ones. And serine and threonine are 2 unique ones in the sense they have a OH group in the side chain. The OH group in the side chain. Okay, so the C beta or gamma, and threonine also has a CH3 group over there. OH and the CH3 group at the gamma position. Okay, so therefore these OH group actually again provide very distinct chemical shifts for the serines and threonine. And tryptophan has an indole ring, and that is the indole proton is again a unique one here. This is the indole proton there. So side chain numbering goes in this manner: alpha, beta, gamma, and epsilon, delta, xi, and so on and so forth. So this nomenclature one has to remember all of this. The tyrosine is aromatic residue once more. So this goes, it also has an OH group in the side chain. Phenyl ring, and then it has a OH group there. Okay, and the valine is again a methyl bearing residue, and it can have it also has very distinct chemical shifts. Now here I show you how the chemical shifts vary depending upon the nature of the amino acid. By and large, you see, this is the proton axis here, goes from 0 ppm to up to 6 ppm, it is shown here. Why I show only up to 6 ppm here? Because we are focusing on the side chains. What are the chemical shifts of the side chains? And the side chains, this starts from the alpha proton onwards, alpha, beta, gamma, delta, and so forth, right? So all the alpha protons appear in this area between 4 ppm to 5.5 or 6. It's all the alpha protons. Look at this here. Look at this. All the alpha protons appear for every amino acid residue. They are in, although there is a small variation there, they appear in this area. Now, 2 of the betas are unique. See, they overlap with the alpha proton area, and that is for the serines and the threonines. This is because they have an OH group in the side chain. Close to the C beta, at the C beta position, you have an OH group. Because of that, the C beta proton is quite close to the alpha proton. Alpha proton is close to the NH2, right? It is alpha C is attached to a nitrogen, C alpha is attached to a nitrogen. Therefore, and the C beta here is attached to an oxygen, it comes close to the alpha protons, that is on the backbone right side. Now, all the other betas are here. Most of the other betas are here, except a few betas which are in this middle area here, the 3 ppm area. And what are these? And these betas are the aromatic betas. Aromatic ring protons create a kind of a ring current, as a result of which these beta protons are shifted quite downfield compared to these beta protons, these aliphatic protons. The beta protons of these aromatic ones appear in the around the 2.5 to 3.5 ppm area. In this area, there are also some other protons, the delta proton of the arginine. Delta proton, why is it here? Because it adds to a nitrogen. Therefore, I said you must remember the structures of the individual amino acids to understand all of these things. The delta proton of arginine, because that is attached to a nitrogen, NH2, NH2, NH2 area. So therefore, that will appear in this area. Similarly, the epsilon proton of the lysine, and this is again attached to a NH3. The epsilon carbon is attached to an NH3, and therefore this appears also in this area. Therefore, this central area is also quite distinct. Centrally, all the alpha protons are here, a few of the distinctly separate beta protons are here, and a few and beta protons in this area are again very distinct because they are connected to the aromatic residues or to the arginine side chain or to the epsilon of the epsilon proton of the lysine residues. Now all other aliphatic ones appear in there. This is the crowded area. See, all the betas, the gammas and the deltas, these are all the methyl protons or the methylene protons on the side chains. They will all appear here. There is one thing which is unique also with regard to the alanine. Alanine beta appears here. This is unique. Alanine beta appears in this area and this has no other things in between. Of course, all other betas are also there. But alanine beta appears most upfield compared to the others. Oops. Alanine beta appears most upfield compared to the others. And the threonine beta also appears in that area. Threonine beta, the methyl group. There is the methyl group is also there in threonine. That also appears. That is called as the gamma. That is not beta. That is called as gamma. CH3 group. So that will appear in this area. All other ones, by and large, the methylenes and those ones are indicated here. This, how does it show up in your NMR spectrum? So here is a two-dimensional NMR spectrum, proton-proton NMR spectrum, chemical shift ranges. What is shown here? The correlations between the backbone protons or the other side chain protons, I mean the exchangeable protons. NH amide, this is the backbone. NH side chain. NH side chain is restricted to what? Only two residues, glutamine and asparagine. Only those two side chains. Of course, arginine and histidine, arginine and lysine also. And the histidine is also coming there. Histidine is coming in this, but that's on the side chain. And then you have here, how do we distinguish the glutamine and arginine side chain? That is because you have two peeps appearing in the same line. Because you remember this is the CONH2. CONH2, the two protons in NH2 are not equivalent. Okay, so they will therefore appear at two different positions and they are connected to that particular carbon which is at this position there. Okay, I am not showing carbon chemical shift here, but which is the proton which is attached to that nearby adjacent one. And now here you see all the alpha protons and the beta protons are present here. Whatever this peptide is not important here, but there are so many residues indicated here. You see the glycines are appearing in this, the alphas are appearing. This is most upfield. Glycine alpha is most upfield compared to all others, and that appears very distinctly in this area. And the serine and serines, you look at this, serine, these two serines, and these are the H alpha and H beta. For example, this one is the H beta and this is the H alpha of serine. Similarly, there is one here and one there, and H alpha and H beta. And now others, if you see, if you look at the leucine, for example, here, then I have here, and what is this line here? What is this line here? This is the water line. Okay. So this runs all the way. This is at 4.8 ppm. So don't look at this peak. So this peak is coming from the water. This is the tail of the water. So you look at this peak. This is the alpha proton of leucine. And then you have these beta protons here, two beta protons, and then you have the two gamma methyls. The methyls are appearing in this area. And let's look at the lysine. The lysine is here, the alpha proton is here, the betas, gammas, and the deltas, and the side chain is appearing here. So epsilon side chain will be appearing at that NH3 will be appearing somewhere else, and that will come here. So this one and this one, these ones are from the side chains of the lysines and the arginines. Okay, that is also indicated from this side. Okay, so it will show. So this is the epsilon proton or the delta proton of the lysine or the arginine. So, and therefore you can see from here the complete connectivity which is seen here. The histidine shows alpha proton, then the beta proton here, and that's it. So there is no alanine here in this. Alanine, okay, this spectrum doesn't have alanine. So the typical certain residues are there in this, and all of them can be identified. And the sequence of peaks which are present, all these are lysines. Okay, these are beta 1, beta 2, gamma 1, delta 2. So that's the way these lysines are going. All the three are lysines there. And why is showing lysine this one here? This is because it is showing a side chain epsilon. It's going to the epsilon there. Okay. So let's move to the next one. Now this kind of a thing which is seen either in the nosy spectrum or in the toxy spectrum. You will see this sort of a spectrum either in the nosy or in the toxy. Now this is the cozy spectrum. And what does the cozy tell you? Cozy we have seen earlier. What all the cozy will tell you? Cozy will tell you about the 3-bond coupling correlations, which is that, so you have this NH proton region here, and this is the alpha proton region here, and this is the betas and the gammas and so on and so forth, right? And this is the aromatic area here. This is the aromatic, okay, and this is the NH to the C alpha area, then the aromatic to the C betas here. So you will also have that sort of a peaks over there. And then once you have that, of course, nothing else will be there on the top. In the toxy, you will have those ones, but in that cozy, you will not have those ones. So you come to these alpha areas, from the alpha to the betas, you will have, and so on and so forth. Okay, so these are the various regions which are classified according to the correlations which are present, A, B, C, D, E, F, G, H. A typical classic example is shown here, an expansion, a blow up is shown here for a particular small peptide. So this actually is a cozy spectrum. What is this area? This is the NH region on this side and the C alpha on this side. NH to the C alpha. And you see how many peaks are there. So you say, okay, well, there are 5 peaks. Therefore, this is a pentapeptide. So all of these are non-equivalent. So I have 1 peak for 1 residue. NH to the C alpha, NH to the C alpha, all of them are there. And this is the cozy spectrum. This will not tell you anything about the nature of the amino acid residue. So you go to the toxic spectrum, which will actually take you into the side chain, along the side chain. So here you see this one, the same 5 peaks are present here. 1, 2, 3, 4, 5. This is 1, 2, 3, 4, 5. Okay, the same peaks are there, and these are the alpha protons, NH to the alpha protons. Now you see correlations further also. Toxic correlations. You have a correlation, for example, if I take here, I have a correlation to this proton, and this proton. And there are 2 protons here. Now you see, looking at this, you go back to the structure of amino acids, then you can figure out what this residue must be. And if I look at this, there is only 1 here, there is nothing else. Alpha to the beta, and nothing beyond the betas. So this will also restrict. There are several residues which finish by beta. And of course, that is a serine is one of those. And there are several residues which finish by the beta only. There is nothing beyond that. Okay, and there are some which finish at the alpha itself. That is the glycines. The glycines finish at alpha. There will be nothing on the top if you have a glycine. Now, if you look at this one there, this has 2 fellows here, 1 here and 1 there. And this typically goes, this is in the area which is beta, and this goes to the kind of a methyl area. So typically by chemical shifts, you can guess this may be a kind of a residue which is like the which contains the beta and the gamma, so like a valine or something like that. So you have the betas here and the gammas there. Or it can be alanine. Alanine can also, but it will have only 1 peak. Alanine will have only 1, it will not have 2, because this is CH3. CH and then alpha proton and then the CH3. It will also. So it cannot be that. So here you have two beta protons here, and these chemical shifts are in the 2.5 to 3.5 area, and you may think that okay, this may belong to kind of a aromatic residue, phenylalanine or tryptophan or things like that. And this fellow shows one here and then two and a few there. So this again is a long side chain, kind of a what is this could be? This is at the 3 chemical shift, and this means that it is something like a lysine. The lysine epsilon proton is there. The lysine epsilon may be coming here, and these are the betas and the gammas, so which are going in that manner. So therefore, these chemical shifts will kind of indicate to you what sort of amino acids there may be present in that in your molecule. Identify the chemical shifts of the individual amino acids. And this is the expansion of the same area in a little bit more detail of the same. And this shows the further expansion of the alpha beta area. So there are further expansions of these ones. There be among the betas and among gammas, what sort of combinations can be present in this area. Okay, so this is the kind of characteristics you will see in your spectra. Now, before we actually start analyzing the spectrum of the protein, it is necessary to have a kind of a dictionary, stepwise how we go and what kind of a spectra we will have to record in order to analyze the spectrum of a protein. So typically, first you record 3 types of spectra. One, there are 2 steps in the process. That what you want to do, you want to identify every proton to a particular amino acid residue along the polypeptide chain, what residue it belongs to and what proton it is in the amino acid residue. So in order to be able to do that, you must have a kind of a dictionary of all the patterns of the peaks which you expect for different amino acid residue. I already shown you the typical chemical shifts of the alpha, beta, gamma and things like that in the chemical shift ranges. But this will reflect itself in the pattern in the 2-dimensional spectra, in the cozy and double quantum filtered cozy and Tox spectra. Some illustration I gave you in the previous slide, and this kind of a dictionary you must have for every amino acid residue. Okay, and then you map this on the experimental spectrum to identify what sort of amino acid residue it may be, and this is called as spin system identification. The first step is spin system identification. Okay, you may call it as the AX spin system or an AMX spin system or the long side chain. Okay, typically all these beta which end as CH2s, these are called as AMX spin system. See, serine, for example, is an AMX spin system. And what about the glycine, which has 2 alphas? Alphas, they can be equivalent, they may not be equivalent. If they are non-equivalent, then it is an AX spin system, the 2 spin system, alphas. So the AMX can be CH2. So CH, and there is another H, let's say H here. So this AMX, so it can form this sort of a thing, these 3 protons. And 2 protons, then nothing else, nothing goes beyond this. So if there is nothing, then or is the OH group here or something for serine. Then one of them is the CH3 group there, which is kind of a threonine. If we have that, you will have different kinds of spin systems which are present, and for different residues, you will have different kinds of spin systems. You have to identify that. The next step will be to connect these individual amino acid residues using the nosy. The nosy give you correlations between one amino acid residue to the next amino acid residue, and that is called as the sequential connectivity in the nosy spectrum. So typically, to start with, when you are given a polypeptide molecule, you record cozy, DQF cozy and toxic spectra. Then you also record the nosy spectra. Shall we take a break here?
Yes, sir.
Okay, son.
थोड़ा उसको मेंशन कर दो.
लिखो, any doubt at this stage?
Any doubt? And wait for 5-10 minutes जो भी है. कोई दाखल
Okay, so let's continue. And now we will see what are the spin systems. Spin systems, these are schematically drawn here. And what, so we are talking about alpha proton onwards, alpha to the beta, to the gamma, to the delta, and so forth. Okay, what is drawn here is, this is taken from Wittrick's book. Okay, those who can have access to the Wittrick's book, this was published in 1986, NMR of proteins and nucleic acids. So in that one, you will see this pictures. Only one side of the two-dimensional spectrum are shown here. The peaks are shown on only one side. They are symmetrically present on the other side as well. So some illustrative things here. These are the side chains of the individual. This is the same slide which was there in the beginning, the first one which I showed you for ready reference. So let us look at the histidine ring here. Where is histidine? Histidine is here, right? Histidine is here. And then you have the alpha pro. This is showing the within the ring. Okay, 2H, and there are two protons, one here and one here. One here and one there. So there is a cross peak between those two there. Okay. Alpha to the beta is not shown here. This is in the aromatic ones. What are the things that are present in the side chain rings? Okay, it's only in the side chain rings. Histidine ring, there are two protons, that shows two peaks, one peak there. Okay. Go to the tyrosine, and where is tyrosine? Tyrosine is here. So there is a proton here, proton there. We're not talking about the C beta, okay? At H beta, we are not talking at this moment. Then you have this one and this one. Typically, these two protons will be equivalent, these two protons will be equivalent. And they are labeled in this manner. Tyrosine ring AA prime XX prime. This is the kind of a nomenclature which is used for, these are magnetically chemically equivalent, magnetically slightly non-equivalent, and that's why you use the terminology A prime XX prime, but chemically they will be equivalent there. So one chemical shift for 2 6 protons, another one is the 3 5H, and you have one cross peak over there in those ones. Now you take the phenylalanine ring, phenylalanine where it is. So we look at phenylalanine, tyrosine tryptophan. Where is phenylalanine in this? Threonine.
Third row, left side.
Here. So you have a proton here, a proton there, proton there, proton there, and a proton there. Now among these again, these two will be equivalent and this will be non-equivalent. So there are three kinds of chemical shifts there. So you will have 3 5, 2 6, and 4. So you will have a correlation from 3 5 to 4 and 4 to 6. Okay. You may also have sometimes this one 3 5 to 2 6 you will have anyway. 3 5 to 2 6 you will have, and 4 to this you may or you may not have. The 4 to this 3 5, that you may or may not have. Okay. Four to 3 5 you have, but 4 to 2 6. So that is here. Which is 2 6? This is 4. 1 2 3 4. So 2 6. This is 2 6. Okay, this is 3 5. You have 4 to 3 5, which is good here. 4 to 2 6. Why it is happening like this?
Loose connection.
Loose connection.
Is connecting this power cable.
So extension board में ना
उसको बोलो
ये पाना है तो वो भी वैसा इश्यू आता है, लूज हो जाता है बार बार.
उसको चेंज करने को बोलो, किसको बोलो, अनुराग को बोलो, जाके रिपोर्ट करो.
Okay. So 4 to 2 6 you may or you may not find, but 4 to 3 5 you will find here, and 3 5 to 2 6 you will find, because these are neighbors, adjacent ones. Tryptophan ring you will see. Tryptophan ring has, again it is.
ये recording problem नहीं होगा ना?
Okay, tryptophan is here. Tryptophan has a proton here, proton here, proton here, proton here, proton here. And then the NH proton, there is the indole ring. So we are talking about, and there is a proton here also. Okay. So and then you will see, therefore there are five groups. 1, 2, 3, 4, 5. There are 5 chemical shifts there. So all of this will be non-equivalent. And you will see this cross peak from 5 to 6, then 6 to 4. These numbers are not, these letters are not very clear there. not written in this manner.
People will understand.
People will
They know
understand
it.
that. Yeah.
In
So
a-
therefore, there are something which are weak, and the ones which are very good, they're strong peaks. They're indicated by full circles. Okay. Now these are the side chains of valines, okay, leucine, isoleucine, and the patterns of the peaks which are given here. So this valine backbone is here. There are two methyl groups from the alpha proton you see to the beta proton. Okay. This comes alpha is here on this side. Alpha to the beta proton, and then from the beta proton to the two gamma protons, this one and this one there, to the two gamma protons here. Okay, that's all you will have there to the beta protons, and there is nothing else. There is no gamma-gamma interaction. That will not be there. So if you take the leucine, alpha to the beta, there are two betas there, alpha to two betas, okay, and then beta to the, this is the beta there, this is the beta 1 and beta 2. From the two betas you see to the gamma, and often the gammas are, the two protons are equivalent. So the gamma to the two deltas. So the two methyls here, these will be non-equivalent. Gamma, there is only one proton, okay? So gamma, there is only one proton. To the two betas to the gamma, there is the single proton there. And from the gamma, you will see to the two methyls, these two methyls. And that is these two peaks there. Okay, so isoleucine also in the similar manner, you will have the correlation from the alpha to the beta proton and the beta to the two gamma protons, which will be non-equivalent here. And from the two gamma protons, you will see to the delta one, which is one methyl. So you will see from the gamma proton to the delta methyl, you will see. Okay, gamma 1, gamma 2 to the delta methyl. These two peaks. Gamma 1, gamma 2. Okay, so beta to the, you will see from the beta to the gamma 1, gamma 2, and also to the gamma CH3. There is also a CH3 there. See this from here, you see to these two, and also to this. This is the CH3 group there. You will see that also, and therefore from the beta, you see three peaks there on this line. So this pattern of peaks which are present here, this will allow you to identify the individual amino acid residues. And these are other spin systems once more: serine, aspartate, asparagine, cysteine, tryptophan, phenylalanine, tyrosine, and aspirin. Now we are not talking about the in the ring. We are talking about what is the alpha beta state. All of these are called as ABX spin systems because the beta 2 beta protons are there. After that, there is no immediate connection. There is no connection to the gamma. And therefore, these are all called as ABX spin systems. There are 7 residues. These are grouped together as ABX or type J spin systems. You see, there are 2 beta protons. Beta to the gamma, there is no this thing. There is no connection. Therefore, it ends at the betas. Therefore, alpha to the beta 1 and beta 2, the two are equivalent, and then you have the beta cross peak. In all of these residues, it is like that only. Okay, alpha to the beta, 2 protons there, alpha to the 2 betas, and then there is no further connection. Similarly here, and there is no from the gamma, there is no proton, and to beta there is, and there is the SH there. So there, in all of these, it is the same. Therefore, all these 7 amino acid residues are labeled as either AMX or ABX. Okay, so you can call it as AMX here, or you can also call it as ABX, depending upon the chemical shift differences between the beta 1 and the beta 2. If they are too close, then of course it becomes an ABX. But if they are reasonably well separated, then it becomes a AMX spin system. Okay, this is the same I had shown earlier, and in the you can see the NOESY spectrum. The NOESY spectrum also shows the same correlations to the because all these within the same amino acid residue, you will have the short distances, right? In the NOESY, you will see cross peaks between residues between protons which are less than 5 angstroms in distance. So you will see cross peaks there. For the same portion of the same peptide there, you will see these 5 things which were present there, and you are seeing correlations to the next C alpha. This is which is this area here. This is the NH to the, this is the NH, backbone NH, and this is the C alpha H. Okay, and NH, you see 2 peaks there. We are seeing 2 peaks. Why do we see 2 peaks? One is you see the same peak which is present for the individual amino acid residue, but you will see another peak. This actually belongs to another residue which is on the left side. So if I say I minus 1, I, residues. So I will show you more details about that. You in the NOE spectrum, you will see also correlations from here to here. NH of this to the C alpha of this. Therefore, you will see pairs of peaks in the noisy spectrum. For every residue, for which one we show here, you have the pairs of peaks, alpha to the beta, alpha to the beta, alpha to the beta. Okay, so you have all of those ones there. Sometimes you may see to the gamma also. So you will see more peaks in the noisy spectrum in general in this area. I'm going too far. Okay. Now, here are certain. Okay. So now this indicates how you actually walk along the polypeptide chain. You make use of COSY and the NOSY spectrum. Okay. As I shown you earlier, the NOSY is showing you peaks for the same amino acid residue, NH to the alpha and also to the previous, and that is indicated here. This is the COSY pattern or double quantum filtered COSY pattern. From the alpha, you see to the two betas there. Okay, it is indicated there. Now this is the backbone here. This is not beta. This is NH. This is okay. This is the NH area. This is also NH area. That's why the diagonal is running like that. The diagonal is running. You have all this here NH chemical shifts, and you see two peaks. To which ones? The NH code. This is in the NOSY spectrum. Okay, this is not COSY. This is in the NOSY spectrum. In the NOSY spectrum, okay, let me write here. These are all NOSY spectra. It is indicated here, of course, in the caption there. The NOSY diagonal of the amide protons. Okay, and if you have a structure which is like this, which is the alpha helix. If you have an alpha helix, what sort of peaks do you see in the NOSY spectra? You see peaks from NH. Okay, now this is NH of residue I. Okay, then NH of residue I plus 1, then NH of residue I minus 1. In between, of course, there are various things. I have only written the NH protons there. Okay, so NH to the two residues on the two sides, you will see. Okay, that means you have to from here to here, you have one further, one behind. And then you go to this backbone, and from here you see this peak there. You come to this backbone, this is the same, and then it ends there. And this one shows to this one there, that is the same as here. And that goes from this to this backbone, and then this backbone goes to this. So this is the way connections are drawn. So half connections are drawn on the top and half connections are drawn at the bottom. This for clarity. In principle, one could have done all of that on the one side only. But this is just for clarity is shown. Some are shown on this bottom and other ones are, because it is symmetric. It's the same peaks which are present here and there. These two are here, these two are here. This is here, this is here. This is here, this is here, this is here, this is here. Okay, the same correlations are there. Half of the correlations are shown on the top and half some others are shown on the bottom. So you will see correlations across along the chain like that. Okay, you go from one residue to the whichever one is next, neighboring residue, you will see those. Why do we see those neighboring residues? We will see soon. That is depending upon the distances which are there. Therefore, this requires a distance information. Now, in the case of a beta sheet, if you have a beta sheet structure, which is indicated here, what sort of a correlations do you see? And that is indicated here. You have, let us say, this particular NH. You will see NH to this C alpha and also to this C alpha. That is NH of residue I. You will see C alpha H of I minus 1 and C alpha H of I itself. Okay, so you will see those. So therefore, you can walk along this considering those two. So every vertical axis, every with two peaks, see two peaks here, two peaks there, every NH. This NH has two peaks, this NH also has two peaks, this NH has two peaks, this NH has two peaks. This does not have. What does that mean? You have come to an end. So that is the end point. So at that end point, of course, there is nothing to go. So therefore, if you identify there is one peak there, then you start from that end. For assignment purposes, you start from that end, then you go to the next one here, then you come down, go to the next one here, go to here, then go to the next one here, come here, next one here, and next one there. So this is how you can walk along the polypeptide chain starting from one point, making use of the fact that if we have a beta sheet, you will have from each NH proton, you have NOEs to the C alpha H proton of I residue and I minus 1 residue. Suppose this is the end point. The end point will not have anything on the other side, right? So therefore, this is the NH of residue number 1, let us say. This is that's indicated on the top here. This is the residue 1, and therefore it has nothing on the top. Its own alpha is present there. From here you go to 2. So 2 has, this is the sequential peak from 2 NH to 1, and this is the self peak of 2 NH to its own. Then you go horizontally, you come here, you come to residue number 3. This is the sequential peak, residue number 2, and this is the self peak of residue number 3. That is indicated here on the side also. You see 2, 4, 3 and indicate. And then you go horizontal here, you come to the sequential peak of residue number from 4 to 3, and this is the self peak of residue number 4. Then again you go horizontally, you come to the NH of residue number 5, and this is the sequential peak to number 4 and the self peak of number 5. So therefore, by this way, you can walk along the polypeptide chain, sequentially connecting the NOEs, which appear in pairs. Okay, so now here is a dictionary kind of a thing, that what sort of peaks you will get in the noisy spectrum. And this depends, as I said, on the distance. Distance between two protons should be less than 5 angstroms. If the distance is less than 5 angstroms, you will see a peak. But intensity of the peak will depend upon what is the actual distance, because it goes as 1 upon d to the power 6. The intensity is proportional to 1 upon d to the power 6. So if the distance is small, you will see a strong peak. If it is large, you will see a weak peak. If it is more than 5 angstroms, you will not see that peak at all. Now, here is an indication of the various secondary structures on the top here. Alpha helix, 310 helix, turn 1, turn 2, turn 1 prime. Okay, these are some other small variations in that one. And the helix turn, helix kind of a motif. And this is for the betas, parallel betas and anti-parallel betas, both are present there. Beta anti-parallel and beta parallel. Let us start from the betas. In the beta, what are the strong peaks you will see? Here is the residue. These are the coupling constants here. Beta below, what is indicated here? This is the coupling constant. We will not talk about that right now. We'll see it later. Okay, next one, next step. And if you see here, d alpha n means NH of I, d alpha n, let me write here. D alpha n means NH of I to C alpha H of I, the alpha n of I. I minus 1. So this you will see it to the I and also to the I minus 1. Okay, so that is the alpha n indicates that. So and then you see this peak is very strong, and this indicates a very thick bar. Thick bar indicates that these peaks are very strong. Sequential peaks. Overview of sequential and medium range NOEs. Okay, sequential and medium range. He is not talking about the self peaks. NH to its own alpha, he is not talking about here. He is talking about which are the sequential one residue to the next residue. That's why I said when I say d alpha n, it is NH of I to C alpha H of I minus 1. And you see, this is a very strong peak for both the beta sheets. Okay. Now, and the NN distance is very small. It is very large. Therefore, this intensity is very small. NN intensity is very small. What is NN? NS means NHI to NHI minus 1 or NHI plus 1, whatever you want to say. And that is very weak. In the case of beta structures, you will see this very strong, this rather weak. Now, if you go to the helix, alpha n I plus 4. What is that? This is the NH of I plus 4 residue to alpha of residue I. 4 residues apart. 4 residues apart, you will see. That is why you see in a stretch of 7 amino acid residues, you will see from 3 to 7, 2 to 6, 2 to 6, and 1 to, there are 4 there, right? This is from 3 to 6, 2 to 6. Sorry, 3 to, there is 7, 3 to 7. 3 to 7, and 2 to 6, 1 to 5. Right? So 1 plus 4. 1 plus 4, I plus 4. So if it is I is 1, then this is 5. So you will see those ones there. And you will see alpha, beta, I to I plus 3. That is beta of I plus 3 to alpha of I. So 3 residues apart. This will be a strong peak. This is much stronger than this. That's why this thick bar is thicker. Okay. In addition to this, you will also see alpha n I to I plus 3. NH of I plus 3 to alpha of I. And you see again, this is a relatively strong peak, but NN I to I plus 2 also you will see, but these are relatively weak. See, depending upon how many residues they are apart, you see the length of this bar is different, because this number of residues is, you consider a 7 residue stretch. In a 7 residue stretch, what are the possibilities that can happen? Okay, so you see here 7 to 5, 6 to 4, 5 to 3, okay, 4 to 2, 2 to 1. So this is the way you can have in this 5 stretch there. And you will see so many peaks in the case of alpha helix. In the 310 helix also you will see those. This one is not so conspicuous, but you see this, and this. All of these 4 you are going to see. And turn 1, again characteristic, different. And alpha helix also you see NN, this is very strong. NN is very strong. Neighboring residues. NN means 1 to the immediately neighboring. It can be on both sides. Okay, I or I plus 1 or I minus 1. Similarly, here also 310 helix also. Therefore, NN peaks are very important for helix characterization, and alpha N peaks are very important for beta sheet characterizations. Sometimes you may see, you may not see this depending upon the intensities of these ones or the dynamics in the protein. You may be able to see, you may not see, but these ones you will always see. Therefore, these are very important to understand determine the structure. Now you go to the turn 1, again this one is very strong. These ones are relatively weak, but sometimes you will see. One has to be aware of this, because you will see when you analyze the noisy spectra, there will be thousands of peaks. Thousands of peaks. These alone will not account for all of those. And there will be many others which are left, and those will correspond to these small intensity peaks. You will have those. So turn 1 and turn 2, both of them have these kind of features. See, 1, 2, 3, 4, you have 2 to 4, here you have 3 to 4. This is a distinction between turn 1 and turn 2. How do we distinguish between these two? You have in the turn 2, you have a strong alpha n peak. In the turn 2, you have a strong alpha n peak. It's not present here. This is relatively weak. There is nothing here. Whereas this one is, you see a 3 to 4, this is NN peak. Whereas in this case, you see strong 3 to 4 and 2 to 3 in turn 2. So you can distinguish between these 3. Turn 1 prime and 2 prime, these are difficult to distinguish between these two there. And helix turn, once again, only have this, but this one will be alpha n will be strong. Half turn, this is not helix turn, this is half turn. So complete turn is not formed, but certain torsion angles on one residue are maintained. So you will see that. So this is the kind of a peak pattern you will get in the noisy spectrum. You should be aware of. These are all. So therefore, these are called as sequential and medium range. Which are sequential? These ones are sequential peaks. These ones are medium range peaks. Okay, because they extend beyond one residue. I to I plus 2, I to I plus 3, I to I plus 4. So these ones go beyond sequential residues. Sequential means immediately neighboring. So these are sequential and medium range proton-proton NOEs. And there is also coupling constant between these. Which are the coupling constants we have? Typically, we have the HN alpha coupling constant. That is we have only one this particular coupling constant, 3-1 coupling constant, NH proton to the C alpha proton. And in the case of beta sheets, this coupling constant is very large. This is about 9 Hertz. Okay, in beta sheets, this is a very large coupling. Okay, therefore you will see these peaks very strong in double quantum filtered COSY or the COSY spectra. So those ones are dependent on the coupling constant. Whereas those ones will be weak in the case of alpha helix. Okay, that coupling constant is very small, 4 hertz. In alpha helix, this coupling constant is very small. So you may not be able to see that peak very strongly in the Cauchy spectrum. Okay, so therefore this coupling constants, and in the turns also there is a variation in the coupling constants there, 4, 5, 7, 5 and so on. So these numbers one has to have so that you can interpret your Nosy and the Cauchy spectra accurately.
Okay.
Yeah.
So in this, let us say tau 1.
Yeah.
You see that DNN,
Yeah.
that line is slightly in one right hand side, it is little bit thicker
Yes.
than the left hand side.
Yeah.
That means
that distance is shorter.
That
Where this is higher,
there's 9-3.
3-4.
3-4 is shorter.
3-4 is shorter than
1-2.
Than 2-3.
Okay, than 2-3. Okay.
Yeah.
So that actually indicates. So everywhere.
Exactly.
Here also.
Same. Yeah, here also it is the same.
2-3 is yes.
Okay, 2-3 is stronger. Yeah, that is so that is this intensity. The bars intensities are proportional to their distance. That is how this is obtained. Okay, now here you see this is from Vitrick's book NMR of proteins and nucleic acids.
So these thin bars, whether whether they appear or sometime may not.
They appear. See, this will depend upon the kind of a mixing time you use. Okay. When you record the Nosy spectrum, if we use a very short mixing time, only the strong ones will appear.
Okay.
If you want the other ones also to appear, you'll have to use a little longer mixing time. So then they will also appear. So by and large, for identification purposes, it is reasonable to use longer mixing time. But when you want to actually quantitate the structure, typically one should use a short mixing time in the Nosy spectrum. Okay, shall we go further?
Yes, sir.
Okay, so now we turn to another kind of interaction. We have talked about this earlier. So this is the so-called magnetic dipolar coupling. This we have discussed in the context of relaxation and NOE, which we talked earlier. And to give a little bit of that mathematics there, that is the magnetic dipolar coupling is defined by this Hamiltonian there. This is gamma 1 for the 2 protons, 2 spins, S1 and S2. The magnetic field is in this direction, BZ, and the distance between the two is R. And gamma 1 and gamma 2 are the gyromagnetic ratios of the 2 spins. Typically, if you're talking about the protons, then anyway, they both will have the same, but it need not be. You can have also proton nitrogen or proton carbon, carbon 13 also can be there. So, or it can even be proton electron. See, electron spin also can be there. So we will see that immediately. Very soon we will see that one of them can be an electron as well. So when you have this, the Hamiltonian which defines the interaction between this is given by this. We need not go into the details of that. This Hamiltonian averages to 0 for isotropic rotation. This is when there is a free tumbling inside the solution, the dipolar coupling gets averaged out to 0. Therefore, it will not result in any splitting of the lines. However, it does contribute to the relaxation phenomena as we discussed in previous classes. So it contributes to the relaxation phenomena. It also contributes to the NOE. And the NOE is proportional to, as I said before, inversely 6th power. Inversely proportional to the 6th power of the distance between the two, and it is proportional also to the products of the two gyromagnetic ratios. Okay, so now in a pure solution, this magnetic dipolar coupling is averaged out. And what is the magnitude of this coupling? Let's have a look at that also. This indicates, the Hamiltonian is written in the same manner here once more. This is slightly in a different format. This is what they are equivalent. In the homonuclear case, this is the Hamiltonian. You have IIZ IKZ minus II dot IK. There are the operators, spin operators. And in the heteronuclear case, the X and Y terms will disappear and you will only have the IZ and KZ. So this is called the secular part of the Hamiltonian. So in heteronuclear case, you will have only this much. And why this is again and again coming this? And the DIK, the constant which is present here, it is proportional to this angle. This angle is what? This is angle, this vector connecting the two spins makes with respect to the z-axis. Z-axis, because the magnetic field is considered to be along the z-axis. Magnetic field if B0 is parallel to the z-axis. Okay, so it is proportional to this angle. Is proportional to this angle in this manner. Okay, so it depends on the distance and it depends upon this angle which is there. Now, roughly, some number, if you have two protons, if you consider, if they are at a distance of 1.5 angstroms, 1.5 angstroms, and this then dipolar interaction is approximately 35 kilohertz. It's a very huge coupling. For proton-proton, it is a huge coupling. 1.5 angstroms if they are. Of course, you may not have two protons which are 1.5. You may be having 2 angstroms or 2.5 angstroms. Accordingly, it will be less. So that is slightly less. Okay, you can control 10 to 15 kilohertz or whatever. So if they are fixed, but if they're tumbling, what happens is this fellow averages out to 0 and you may not be able to see those. That this angle is completely flexible. It goes around anyway. It can go from 0 to 360. When you take an average of the average ensemble average over this term here, 3 cosines. There is square theta. Yes. It's just 3 cosine square theta. Cosine square theta I minus 1. So this term actually goes to 0 when you take an average of this. Okay. So, however, under certain conditions, this may not get averaged out to 0 and the residual may remain. A residual may remain. And that is called as the residual dipolar coupling. The residual dipolar coupling in a solution. How does that happen? Under what conditions does it happen? This happens under these conditions. Suppose you create a bicelles. These are all made of the lipid bicelles. Okay, so these have ordered species. These are made of lipids. They are very ordered species. You take a certain concentration of that inside your gel, inside your solution. Then what happens is the free rotation of these molecules is hindered, is partially hindered, not completely hindered, but partially hindered. So as a result of this will not get averaged out to 0, but the small residual dipolar coupling will remain. And that residual dipolar coupling will not appear in your spectrum. So instead of getting a J separation in an isotropic case, between when there is a splitting, suppose you are talking about the NH proton, NH, coupling from this to this. If you are talking about this coupling, this is typically about 90 hertz, 95 hertz. Okay, this is a one bond coupling. Okay, and the dipolar coupling between these two will vanish in the case of isotropic solution. Okay, it's come free tumbling. If it is not free tumbling, it is restricted in motion like this, a residual dipolar coupling will remain and that will appear along with the J, and you see this is what will happen. Two peaks which are separated by J, now they will be separated by J plus RDC. Isotropic here, J plus RDC. RDC can go negative or positive, both, depends upon which way it is, orientation, whatever it is. It can decrease or it can increase. So what is the consequence of this? So you can measure this RDC. So what is the use of this? So you see, for example, here in this is 93.38, 95.27. So a small thing here. And it can vary from 10 to 15 hertz. Even you will be able to get this sort of a variation. Now, these are certain examples of those here. So this is isotropic solution. The decoupled spectrum, completely decoupled spectrum. Okay, 3 residues which are present here. And this is the situation when you have a isotropic solution decoupled to nitrogen. Isotropic solution decoupled in proton and nitrogen. So both the couplings are removed. So this is the nitrogen chemical shift here and this is the proton chemical shift here. Okay. And what is this coupling showing here? This coupling is the nitrogen proton coupling. Nitrogen proton coupling is shown here. Okay. And this is all 92 hertz. It is all the same. One bond coupling. This is all one bond coupling. It is the same always. Okay. In the isotropic solution. Now, this is the anisotropic solution as indicated here. In anisotropic situation, see this coupling is seen as 106 hertz. This is seen as 87 hertz. This is seen as 99 hertz. See, all of them are different. Okay. So therefore, from here you measure what is the value of the RDC. The value of the RDC you get by measuring this and measuring this. So difference of those will give you the RDC. Oh, this is finished here. Okay, go to the next one.
This is the last slide.
And this is the last slide.
So
Next one. 4. So this is the structural parameter. So why is it a structural parameter? Because it tells you about the distance between the protons, which are how far the protons are there, and it will allow you to estimate the distance, and that provides a very useful structural parameters. I will show you the application of that in this as we continue the discussion of the structure determination here. So what we show there is the parameters which are there, the distance parameters. Then from the RDC you get a residual structure, residual dipolar coupling, and that is related to the distance. How much is the distance between the two protons? Okay. Typically that residual distance is approximately about, I can go longer than NOE. It can go to some 10 hertz, 15 hertz, that, because it goes to the cube. So you will see that. Now let us see how to use all of these inputs for structure calculation. So this is the ultimate for us, right? Protein structure determination. Okay, so we use all of these experiments. We look at the NOESY, and the NOESY we have, as I said, various kinds of correlations which are present there. There are some which are sequential NOEs, which are long-range NOEs, and the long-range NOEs when the protein chain folds in a particular manner, and you will have two protons coming close by, and that is, so here this is rather is not such a great spectrum, but it was one of the very first spectrum. That's why I'm showing you this here. But today's spectra are far more complex than this. Much more a larger number of peaks you see. But this was one of the very early ones, which is therefore I should give importance to that. That's why I picked up this one there. So here, apart from the sequential NOEs which you have, suppose the polypeptide chain is like this, A, B, C, D. There are 4 protons which are at distinct positions there. So you have the D is here, A is here, B is there, and C is there. They are at different positions on the polypeptide chain, and they also have different chemical shifts. Okay. Now, if the polypeptide chain folds in this manner, these protons come close by. If they come to close than 5 angstroms, then you will see a cross peak between them. If the chain folds like this, these ones are coming close by, they will see a cross peak between them. And likewise here, these will see a cross peak between them. The first step, therefore, has to be obtain sequence-specific assignments. Okay, this is the next step towards the structure calculation. We already seen how to obtain sequence-specific assignments using the COSY and the NOESY spectra. We already seen how to obtain sequence-specific assignments, sequential NOEs, and the spin system identifications. We are going to the next step now to identify the long-range connections. The long-range connections will tell you about the three-dimensional structure of the protein, how the protein folds in this manner. Accordingly, you will have different kinds of correlations. Therefore, the NOESY spectrum in toto is called as the fingerprint of the structure of the molecule. Whereas the COSY, the double quantum filtered COSY or the TOXY, these are called as the fingerprints of the spin sequences, the polypeptide chain, amino acids, primary structure. Okay, now having got that information, you have two inputs now. One is the distance between the protons, the various protons, and then the second input is the coupling constant. We have only one coupling constant, the NH to the C alpha, NH to the C alpha H, and that determines the torsion angle phi. All right, the NC alpha bond, and then the proton-proton coupling is related to phi. We have already seen how that varies, the alpha helix, the beta sheets, and turns and things like that. So you have these inputs. You have the various distance inputs from the NOE quantification. And also, of course, as I said before, the NOE is proportional to the inverse 6th power of the distance. You quantitate the NOE intensities, and then you will be able to estimate what sort of a distance it must be. Okay. Now, okay. So that is from the NOEs. Now there is one more parameter which is, okay, I already talked to you about the residual dipolar couplings. That also gives you the distance information. Now there is one more parameter which is called as the paramagnetic relaxation enhancement. This is present when there is an electron, unpaired electron in your molecule. Unpaired electron can happen either because of intrinsically some kind of manganese is there or something like that is there in your molecule, or you have attached a spin label to some particular residue in your polypeptide chain. And suppose their spin labels are like MTSL and such kind of spin labels are there. These are small molecules and they have an unpaired electron. And that unpaired electron can be attached, if that label is attached to the protein, then what happens is that unpaired electron also has a magnetic moment. So it will have a dipolar interaction with the nuclei. And the dipolar interaction causes the relaxation. Okay, so we have seen the proton, we have already seen if there are proton-proton interactions, then you have the interactions between this residue and this residue. Dipole-dipole interactions cause fluctuating dipole moments, fluctuating magnetic fields, and that leads to relaxation. Here in one case there is a nucleus, other one is a electron. If there is an electron, the electron is completely jumping from one state to the other state. Okay, and the molecule is tumbling also, and it is also jumping from alpha to the beta state in its electronic state. Now, therefore, this jumping around influences the relaxation properties of this nucleus. And notice what I said before, the dipolar interaction depends upon the product of the gyromagnetic ratios. There's gamma 1 into gamma 2. But in this case, the gamma of the electron, or G factor, this is 2000 times larger than that of the proton. Therefore, it has a very major effect on the relaxation properties of the nuclei. And as a result, the distance information what you can get from this relaxation effect of the paramagnetic species can go to much larger numbers. It can go to about 15 to 25 angstroms. That long range distance we can measure if we can obtain the paramagnetic relaxation enhancement. So typically for this one has to do certain kind of synthesis that you have to attach a spin label to a particular amino acid residue. Often one attaches this to the cysteine residues. The SH residue, which is their SH group in the cysteines, at the SH group you attach the spin label. Okay, and from that spin label you see relaxation influences on the other portions of the protein, and from that you calculate a long range distance. So therefore this is an additional structural input you have, and the distance is related to this parameter here, which is the paramagnetic relaxation enhancement. It is of course related to the correlation time, the tau c, which is the tumbling rate of these ones, and this is typically characterized in this manner. Once you have this number, this is the constant, so you can estimate the distance by this, taking the 6th inverse power of this, and you can get this distance. You get a long range distance here. So therefore, all of these contribute immensely to the accuracy of the structure calculations. The larger the number of constraints you will have, the better it is for the accuracy of the structure determination. Okay, having got all of these distances, now you have to build a model. To build a model to calculate the structure, you start with any random structure in the model. Polypeptide chain you have, start with a random structure and put in all your distance constraints. Did I have another slide before? I missed that. So you have here various kinds of distance constraints, right? So this is the lower limit and this is an upper limit for the distance. How do you get two limits? You get two limits because your certainty, there is a certain amount of uncertainty in your distance estimation. Okay, you cannot define the distance so precisely as it is 3.45678 angstroms. You should not do that because your quantification is not that precise. There are also experimental parameters like mixing time, dependent variations, all of this. Therefore, you always is safer to define a range for the distance. It must be in this range of the distances. And this can be narrower, it can be wider depending upon the intensity of the peak. And then you define an energy function for NOE. EF is the standard steric factors and all of those bond energies, bond angles and all of those ones. And ENOE is defined in this manner. They define a potential function like this. So if there is a deviation of your in your model, so this is the structure, this is the distance in your model, Rij, okay, or Rmn. This is the distance. If there is a violation of the lower bound of the distance, that means it lies somewhere here, then this is the potential function that will apply. If it is somewhere here, then this is the function that will apply. Is a violation on the upper distance, violation on the lower distance, or violation on the upper distance. So you add up all of those ones, okay, in your structure and calculate an energy function. These are the force constants for those. These are harmonic oscillators, potential functions described. You calculate this energy, okay. Now this energy has to be zero. If this condition is satisfied, if Rij is equal, then you set this value equal to 0. If this is within this range, if the distance is within these limits, then the energy value is 0. That is here indicated. If it is deviating from them, then you have this potential function. If it is within the range, then the value is 0. Therefore, you need to optimize your structure so that you get a value 0. But of course, by and large, you don't get a value 0, you'll get a small minimum number, as small as possible you can go. That is the convergence, how much you can converge, and depending upon the level of convergence, you may get more than one structures. That's why you will see in the NMR structures, you have a overlay of some 5-10 structures displayed, and that defines about how much convergence you have achieved in the calculation of the structure. If it is very well defined, then your potential, this is called the target function. If it is extremely small, then you have a very well defined structure. So you will not have too much variation in this. Okay, now that you will immediately see in the next one there. So here you start from an arbitrary structure using an algorithm called as distance geometry. 6 different starting points are used here. Why do we do that? Because you need to be sure that your starting structure does not influence your final structure. Your constraint set is sufficiently good so that you get a unique structure. So therefore you have to start from various different ones. Okay. The illustration is taken 6, but typically you may want to do with more than that. Okay, and then you put in the constraints and optimize the structure on our computer. Optimize meaning what? You have to keep changing the torsion angles, alpha, beta, all of these 5-side torsion angles, you have to keep changing. And then you calculate and observe how your target function or the potential energy is changing. And when it is converged, then you get this. So in this case, you see, these the end points here are these are stereo pictures. Okay. In all of these, you see, although the initial structures are different, you have the same structure at the end, which means your constraint set is sufficiently good and you have a unique structure determined from the NMR data. Okay, now that was the distance geometry algorithm, but there is another method to obtain the structure, and that is called as a restrained molecular dynamics simulation. Now what you do here is, you heat up on your computer, okay? You heat up their molecule to very large temperature, something like about 2000 degree Kelvin, very high temperature heat. What is the purpose? You don't break the bonds. You heat it up so that the chain is completely opened up, completely random, okay? And this completely fluctuates there in the open manner. Then you put in the constraint and start cooling the system on the computer. Start cooling the system means your degrees of freedom are getting restricted slowly. The motions are becoming restricted, motions are getting slow. And as the bonds are getting formed, the motions is slowed down further, the temperature goes down further. So you slowly lower down the temperature, and eventually you see when you come down to the room temperature, you see the change is extremely small. The structure gets converged to a particular kind of convergent structure. So this is what you will get in the end. This is the stereo picture of the two, and these are the intermediate ones. Either 4 picosecond, 8 picosecond, 12 picoseconds, and 30 up to 30 picoseconds it is shown. By 30 picoseconds of cooling, the molecular dynamics simulation, the protein has folded into a reasonably native structure. Okay, this is what we get. And you see multiple overlays here. This is, as I said earlier, this defines on the accuracy of your distance constraints, how well your distance constraints are. If they are very good, then you will find all of them exactly on top of each other, but rarely you will find that exactly on top of each other. Some portions may be very good, some portions may be poorly defined, and that can happen. This typically it happens in the case of loop areas. When there are loop areas, the structures are not so well defined because the NOEs are not so well defined. There is more dynamics intrinsically in that one. The NOEs do not represent the distances so quantitatively. So in that situation, either you will have a smaller number of NOEs or you will have imprecise distance estimations. So you will have kind of fluctuations there. Nothing. What is happening? Now I move it, I'm only getting this writing, it's not moving the slide. Okay. So at the end of that, what you have to see now, having obtained the structure, you should see whether it is correct or not. What is the test? The test for that is, oh, we have actually run out of time already.
Yeah.
The test for that finally is the Ramachandran map. All yours, after you got the structure, you have to estimate the phi psi values. The phi psi values have to lie in this allowed region of the Ramachandran map. Once you have this, then your structure is proper and well defined. I think this will not get finished.
Yes, it is not. I think you can conclude it now
I
and
think we'll stop here and go continue in the next
class.
next class. This is how many slides are over now?
4, 5.
It's
Oh, this has 35 structures.
Yeah.
It will take long time.
To take. I think 41 slides are there.
40.
Okay.
Okay, next class.
Okay, sir.
I think it is better to understand it.
Yeah, yes.
Going
When you are putting this nitroxyl radical,
Ah.
so it doesn't change the lines steps.
It does, it does, it does. We will see.
So you are getting some
Line bond-
information somewhere.
Yeah.
You have to compromise
Line broadening you will get.
line broadening.
Line broadening will happen. Of course, your intensities will go down. But that is an indication.
Okay.
That is an indication that it is interacting
Yes, yes.
with that.
Yes, good.
Okay, we'll stop here
Yeah.
and
I think no question.
Yes.
So next class will be on
Friday.
Friday 3:30. Okay, that circular will be going.
Yeah.
Yeah, thank you.